Good afternoon, everyone. My name is Dr. Evan Norris from the University of Colorado, working closely with Dr. Jeff Mackey. I am pleased to present some exciting updates on the infamous pig study, which is finally coming to a conclusion. Here are our disclosures. Now for a bit of background. Recent in vitro work has described the nonlinear relationship between the blood concentration of gadolinium-based contrast agents and R1 as well as R2 star. Thus, arterial signal intensity in contrast-enhanced MRA is expected to rise, plateau, and ultimately diminish with increased gadolinium concentrations. This suggests that faster injections will not necessarily increase signal intensity, but instead shorten bolus duration with resultant blurring and image degradation. While theoretically based, there currently does not exist a validated model whereby contrast-enhanced MRA signal intensity can be accurately predicted based on in vivo gadolinium concentrations. We thus seek to establish such a model based on the in vivo behavior of gadolinium in a pig model. These were our methods. 4 gadolinium agents were investigated on 8 juvenile German swine. I know we all love the generic names, but I'm just going to stick with the brand names for the purposes of consistency. We investigated ProHance, Multihance, Gadavis, and Ablavar. 2 pigs each received a total of 3 injections of the same agent in randomized order, either at 1, 2, or 3 milliliters per second. Arterial blood was then sampled every 2 seconds and sent for mass spectrometry to determine the precise gadolinium concentration. This was then used to predict MRI signal intensity and compared with the observed signal intensity from concurrent high temporal resolution contrast-enhanced MRA at 1.5 Tesla. Prediction of signal intensity came from the following equations below, which were derived by Wilson et al. Taking into account protein binding, the shutter speed effect due to water flux across the red blood cell, and the R2 star lengthening effects in blood. Moving on to results. Here are the results of the 3 consecutive injections between Multihance and ProHance. Multihance on the top row, ProHance on the bottom row. The red line denotes actual signal intensity by time-resolved contrast-enhanced MRA angiography. The blue line is the prediction. Take note of the excellent matching, both in the baseline and in the tail. Here are further results for Ablavar and Gadavis. Ablavar on the top, Gadavis on the bottom. As you note, throughout injections 1, 2, and 3, there is also excellent correlation between the predicted and the observed signal intensity, especially noting the baseline and tail. Here we aim to show the differences between predicted and observed signal intensity based on the inclusion and exclusion of different variables. On the left, we demonstrate the effects of predictive modeling using both R1 and R2 star in blood. In the middle, these graphs highlight just using R2 star modeling. In other words, we ignore R1 corrections for blood and assume that R1 behavior is as if it is in plasma. On the right, we ignore both R1 and R2 star modeling. For these pigs imaged with ProHance and Ablavar, correlation between predicted and measured signal intensity was excellent when employing both R1 and R2 star, the graphs on the left. These relationships broke down and became much less accurate if R1 modeling or both R1 and R2 star modeling were neglected, the graphs in the middle and on the right. Results were similar between Multihance and Gadavis as well. In conclusion, the sublinear behavior of R1 and the high R2 star values in blood due to red blood cell water exchange and the effects of excluding gadolinium from ellipsoid red blood cells respectively, as described by Wilson et al., are demonstrated to hold up in an animal model, thus validating this theoretical and experimental work in vivo. This implies that the relationship between gadolinium concentration in blood and signal intensity can be accurately predicted enabling for more accurate modeling that can optimize the use of gadolinium-based contrast agents for contrast-enhanced MR angiography. Thank you for listening, and thank you for your time.